Ladies and gentlemen, please sit down. Ladies and gentlemen, we are going to listen the opening speech represented by Vice Dean to Faculty of Animal and Agricultural Sciences Universitas Diponegoro. Ladies and gentlemen, please welcome Agus Setiadi, SPT, MSc, PhD.
Thank you very much, moderator. Assalamualaikum warahmatullahi wabarakatuh. Good morning, student and all participants, and of course the guest lecturer today, Professor Elizabeth Atkins, who is joining us by online by Zoom meeting. We are welcoming you to the invited lecturer series organized by Agroekoteknologi Study Program, Faculty of Animal and Agricultural Sciences, Universitas Diponegoro. And thank you very much for your participation in this event. I'm sorry today our Dean, Professor Bambang Paluyo, cannot join and open this event because he has another activity in Batang. First, thank you to the Honorable Rector, Professor Yos Johan Utama, who always support us. So the invited guest lecture series can be conducted today. Second, of course, I thank to the Professor Elizabeth Atkins, the Professor of Plant Pathology, the University of Queensland, for taking part in invited lecture series. Faculty of Animal and Agricultural Sciences today is the reputable faculty in Indonesia and of course in the world. Now we have 7 study programs, start from Bachelor of Animal Science, Bachelor of Food Technology, Bachelor of Agroekoteknologi, Bachelor of Agribusiness, Master of Animal Science, Master of Agribusiness, and Doctor of Animal Science. And Alhamdulillah, all of the study program now got the excellent accreditation from the accredited Indonesian government. And 3 of our study program now also already received international accreditation from the ASEAN. We have Bachelor of Animal Science, Bachelor of Agribusiness, and Bachelor of Food Technology, now already accredited by international accreditation ASEAN. And based of our QS World University rank, our faculty now managed in 400 plus rank in the world in the field of agriculture. So Alhamdulillah, in Agroekoteknologi also can support the rank of agriculture field for our faculty. And for the QS graduate employable rank, put Universitas Diponegoro in the first place. And usually every year we holding our annual event, which is we call invited or visiting professor. And today the topic is morphology and genetic diversity of Fusarium Oxysporum Cubense. And this is very interesting topic, which can build our student and our lecturer also, based on knowledge regarding the plant pathology and also the integrated pest disease management. So we are all thanks to you, Professor Elizabeth Aitken, which experience regarding the plant pathology, and also related with integrated pest management. And of course, the student, all of the student and participant can use this event, maybe you can also much learn about the plant morphology, and I give my deepest gratitude, of course, to the all committees who have directly and indirectly supported the success of this invited lecturer program. Although we try to do best, but maybe I also apologize. Should there be inconvenience that maybe occur during before, during, and after the event. And with these words, on behalf of Faculty of Animal Agricultural Sciences, I declare the invited lecture series today open. May God bless you all and stay good health, and to make this event a successful and enjoyable one. And thank you very much, and enjoy this invited guest lecture. Thank you very much. I give to the moderator. Thank you.
Okay, thank you, Agus Setiadi, SPT MSI PhD. And now we will hold a photo session. To speaker and participants are welcome to turn on camera and take a position. We will count to start the photo session.
Good. Start. Right. Okay. I'll share now.
Okay. We will count to start the photo session.
Wrong one.
1, 2, 3. Okay, thank you. We are now continuing our meeting to the main agenda about the topic Morphology and Genetic Diversity of Fusarium Oxysporum f.sp Cubense, which will be delivered by Professor Elizabeth Aitken from the University of Queensland, Australia. The lecture will be led by Aisyah Surya Bintang, SPMSC. Ladies and gentlemen, please welcome Professor Elizabeth Aitken and Aisyah Surya Bintang, SPMSC.
Okay, thank you very much for MC. Okay, good morning, ladies and gentlemen, Honorable Vice Dean 2, Honorable our invited speaker, Professor Elizabeth Aitken. How are you, Prof. Liz?
Good.
Okay, how's the weather in Queensland right now?
It's very, very pleasant today. We haven't got the rain. There's a lot of rain further south in New South Wales, but we're still okay at the moment. Thank you.
Okay, we really hope that you are safe over there. And before we start, I would like to express my gratitude. Thank you very much for your kindness and consideration to become one of our speakers today. My name is Aisyah Surya Bintang, but you just can simply call me Bintang, and I will lead the lecture and discussion for today. And Fusarium Oxysporum f.sp Cubense, or later I will say it as FOC, is the causal agent of Fusarium wilt, or Panama disease, which is the most serious disease affecting the whole banana industry. FOC is one of more than 100 forms for my specialists of the Fusarium Oxysporum species complex of pathogenic as well as non-pathogenic fungi. This pathogen is commonly spread locally, nationally, and internationally in infected rhizomes or sucker and attached soil. Fusarium wilt is a lethal fungal disease of banana. So today we invite an expert about Fusarium Oxysporum f.sp Cubense, so we can get more knowledge about morphology and genetic diversity of Fusarium Oxysporum f.sp Cubense. Professor Elizabeth Aitken from the University of Queensland, Australia. So maybe I will read some CV or brief introduction about Professor Elise. Okay, so Professor Elizabeth Aitken, she is professor in plant pathology, school director, research in the University of Queensland, Australia. So you can see her profile, okay, and her contact. Okay, so her teaching and development about plant biology, fungi and plant diseases, plant microbe and insect interactions, and also about molecular diagnostic and plant protection. So her educations, she finished BSc Honors in Agricultural Sciences, Edinburgh University, and PhD studies in plant pathology, Aberdeen University, and postdoctoral studies in plant pathology, Birmingham University, and the Sainsbury Laboratory. Okay, so next about her research and journal publications, mostly about Fusarium Oxysporum f.sp Cubense, and not only about Fusarium, but also about another fungal disease. I also read about Ganoderma. She did a lot of research. She has a lot of publications and a lot of books and many more. Maybe you can find her publications on the online. Okay, thank you. So, Prof. Liz, please
Yes.
give the lecture and we will have the discussion later. So please, time is yours.
Thank you very much, Bintang, for your introduction. I'm going to try and share the screen now. Please let me know, just check that I'm opening the right file, and I should put it into presentation mode. Try this one. So are you seeing that slide in presentation mode?
Yes.
Right, okay.
Very well, thank you.
Right, thank you. So it's gone to my alternate screen, so I will be a bit of a side view here, if you don't mind. So I've been asked, and very kindly, thank you very much for the invite, to talk about some of our work looking at the diversity of Fusarium Oxysporum f.sp Cubense. Now, sorry, it's speeding away with me here, unfortunately. I'll go back. And I'm in the School of Agriculture and Food Science here at the University of Queensland, based in Brisbane. The university has a couple of campuses. I'm at the St. Lucia campus, and it's a rather nice building with an old central courtyard. Unfortunately, I'm not in one of the more attractive buildings, but this is a view of the campus here. It's in a bend of the river. Unfortunately, that means we are sometimes prone to flood, but hopefully we're crossing our fingers for this year. And we're relatively close to the central business district. Now, why I put this map in, this aerial view in, is because this place over to the right here called Eagle Farm is where the first description of Fusarium wilt on banana was made. It was a medical doctor who was a keen horticulturalist, noticed on his bananas that they were starting to wilt, and he sort of looked at it and identified that it was a causal agent. Well, I don't know if he actually was able to identify the fungus completely, but he certainly described the symptoms. Now, that's not to say Fusarium wilt comes from Brisbane. It was just that somebody with the ability to study it encountered it here. And here in Australia, we do have several races of the Fusarium wilt fungus. We have what we known as subtropical race 4 and race 1. Tropical race 4, it has occurred in northern Queensland, where most of our bananas are grown, and it's also in the Northern Territory. Now, why I mention these three, because it comes in different ways. Now, there's a bit of a history to Fusarium wilt, and we'll look at some of that now. So the main players, I'm just mention people before I forget them at the end. These are a combination of postdocs and research students who worked in the lab who contribute to the work I'm going to discuss today. So what I want to talk about is a little bit about banana cultivation, talk about the pathogen, the interaction with the plant, and then talk about the host itself. Now, I realize it's, correct me if I'm wrong, we've got quite a varied background of people listening in today. Some who are animal scientists perhaps and aren't so familiar with plant diseases, and up to different levels. So if you need any explanation, please just interrupt. I'm happy to expand on any area. So just looking, and then on the other hand, there might be some things I'm talking about that everybody knows about. So forgive me if I do that too. So looking at global banana production, and by banana, I mean muses, things includes both cooking banana, plantain, and dessert banana. So if we look at various statistics, I mean, they vary all the time, but this is just a snapshot of one. We find that the biggest producers of banana are India and China, but they're mostly servicing a domestic market. And then Indonesia, you're quite high up there in your banana production there. And why I mention Uganda is because we're involved in a project that's funded by the Gates Foundation, and it's looking at subsistence banana production in Uganda and Tanzania in East Africa. So, and Australia, we're quite far down the list when it comes to banana production, but most of our banana production is for a domestic market. We don't export, and on the same token, we do not import banana. So we grow our own bananas here. And this is just a snapshot of an image taken in the market in Uganda of how prevalent bananas are. And I'm sure if you went to some of the regional parts of Indonesia, you would find similar things, but different cultivars. Bananas are consumed across the world, but not necessarily produced across the world. And the markets of, particularly of North America and Europe, the consumption there really stems from what happened in the early parts of the 20th century onwards. Prior to that, prior to the advent of steamships, they really didn't have much access to banana. Australia did because we're in a more tropical region, but generally much of the Western countries weren't really exposed to many banana because they couldn't get them to market in time before they ripened. And that plays a significant role in the spread of Fusarium wilt, as I'll mention. So just to look at, excuse me, in East Africa, they have two main types of cooking banana, matoke and mashari, and they eat banana with everything, with every meal, and they make various things from banana juice and even banana beer. But what this image in the middle illustrates is a program through the International Institute of Tropical Agriculture and also some of the national breeding programs in East Africa and Tanzania, is that through breeding, they've actually increased the yield of some of these local varieties. So that's just to put in the thing that bananas can be bred. Much of us think that, you know, it's a sterile crop for most of the time, it's polyploid, it's difficult to breed. Banana improvement has been made and is continually being made, albeit that very recently. So as I mentioned before, in the early to mid part of the 20th century, that's when you started seeing a big export trade in banana. And you've got a lot of bananas were grown in Central America, and most of these focused on one cultivar known as Gros Michel or Big Mike. And Fusarium wilt had been identified, as I said, back in 1876 in Brisbane, and it was sort of ticking along very quietly without making much impact at that point. And it was only in the middle of the 20th century that you started seeing an upsurge in incidence of Fusarium wilt. And that was because, unfortunately, the fungus had been introduced into Central America. And it was exposed to a cultivar, a Gros Michel, a single clonal line that was susceptible to Fusarium wilt at the time. And it was particularly problematic in Panama, and that's why sometimes you hear it being referred to as Panama disease. And it's a soil-borne pathogen. It's a fungus. It's in the soil. It enters the plant via the root system and then moves up into the vascular tissue, into the water-conducting vessels of the plant, the xylem, and it physically clogs the xylem up. The plant wilts because it can't do its normal water transportation, and the plants collapse and without producing any fruit. Now, why it was important in Central America is because, as I mentioned, the steamships had started becoming appearing at the beginning of the 20th century, and they were using these steamships to ship the bananas back to the markets of Europe and North America. And many of the steamship companies actually got into the way of growing bananas themselves. And when they got in their land, got contaminated by the Fusarium wilt fungus, because the thing is with this fungus, once it's in the soil, you can't get rid of it. And when this land got contaminated, they'd start purchasing more land. And without the knowledge of being plant pathologists, they took their bits and suckers of the diseased banana plants to their fresh properties, and the disease spread from there and there. And to the point that, you know, the whole industry collapsed. And it was only with the use of someone pointing out that Cavendish cultivars were resistant to this fungus that they started replacing Cavendish, Gros Michel, and we see that Cavendish is the main cultivar of trade today. Okay. So, just to sum up, Fusarium wilt was first described in Brisbane in 1876. There was numerous reports in tropical America in the 1890s, but it was only in the middle of the 20th century that you really got it taking off and causing this problem. It's a root-infecting fungus. It enters by the roots, gets in through the rhizome, up through the xylem, and causes a wilt, leading to death of the plant. So often what you'll see is this sort of collapse of the leaves, the yellowing of the whole leaf, as opposed to a leaf pathogen that you see necrotic spots and things. So the whole leaf collapses and you get the skirt around the growing point. So the first wave was in the mid-20th century when Gros Michel was the cultivar of the trade was then susceptible to it. Meanwhile, bananas in subsistent areas in where bananas or local bananas are growing weren't necessarily affected to the same degree. So the issue was mostly in the export trade at that point. So, so this led to the rise of Cavendish. It became the cultivar of trade, and it's resistant to Fusarium wilt at that time. So just a little bit about Fusarium Oxysporum. Fusarium Oxysporum is probably an asexual fungus belonging to the Ascomycota. So it's a member of the higher fungi, the Dicaria. It doesn't produce a sexual stage. So we just see asexual spores. We don't see it going through a meiotic stage. Fusarium Oxysporum as a species, or so-called as a species, has a very wide host range, but it exists in host-specific strains. So we use the term formis specialis, meaning host-specific. So the strains that will attack Cavendish won't attack other things. It can also be an endophyte. It can live in the roots of other of various plants, of weed species, and we can take the pathogenic strains of banana and we can find them in the roots of weeds in the field amongst the banana plant without causing any effect. It can also be a saprophyte. So many fungal species that are plant pathogens are generally poor saprophytes. They can't compete in a saprophytic environment. Unfortunately, Fusarium Oxysporum can, and it's a very successful pathogen as a consequence. So if we look at it, we see Fusarium Oxysporum f.sp strains that will attack tomato, and it can be quite a problem in the tomato industry. And we call those ones, excuse me, Fusarium Oxysporum f.sp Lycopersicii, or FOL for short. We can see it a problem in strawberries, and we call that one f.sp frugeri, in ginger, in zingerberry, in cotton, cousin basinfectum, and in banana, of course, and that's Fusarium Oxysporum f.sp Cubense, or FOC for short. So in banana, the symptoms that it can cause necrosis at the leaf margins, it can cause leaf discoloration and eventual complete chlorosis. It can cause the stem splitting. So it's where you look at the base of the pseudostem and you see the sort of splitting away, and petiole collapse there. But to me, the most diagnostic, because banana plants can suffer from these things from other causes, whether it's fungal or not. But the most distinguishing feature is when you chop the pseudostem down and you see this characteristic vascular discoloration. And that's associated with the plant's response to the fact that there's a fungus in the xylem vessel, and it's reacting, it's producing phenolics and trying to inhibit that. You see that sort of vascular discoloration with other vascular wilt pathogens, even in trees suffering from ceratocystis and things like that. So banana cultivation, for those, I mean, I'm sure most of you are all familiar with banana cultivation, but just to emphasize the point, how it's done in traditional ways is by going and taking the suckers and from the rhizome and little developing buds on an existing pseudostem and breaking them up and planting them, and subsequently doing this the next season, taking other suckers and rooting them to other fields, and you're multiplying it up by traditional bits and suckers. And so you can imagine if you're dealing with a fungus that's root invading, it's very persistent. Once it gets into the tissue, it's persistent in soil and roots, and you use these little suckers as your growing point. You go to new fields, new parts of the field, new plantations, it's going to spread incredibly. And that's what happened. That's what happened with what we now know as race 1, which caused the epidemic on Gros Michel. And unfortunately, that's what's happening with tropical race 4. So if you're going to initiate from bits and suckers, and you might be going in as a farmer and not realizing if you've got Fusarium wilt, because it might be early stages, but the fungus is still there, you spread it like this. And it's unfortunate in some places, and that's how effectively it's spread. I visited China a few years ago before the pandemic, and I could see areas that had banana in it, and it was spreading, and they were basically replacing it with other crops. They couldn't plant anymore because it spread that much. So tissue culture derived plants are a better option because, and I'm sure many of you are maybe familiar with this, by taking plants into tissue culture, you're eradicating most of the pathogens. We still have to be very careful with viruses, that we make sure you've got virus-free material that you're initiating from. But you're growing them up in tissue culture. If the Fusarium was there, it would come out in the media and it'd grow all over the media, and you know it was there, so you wouldn't initiate from such plants. So you can multiply up, and it's all clonal, as banana always is clonal, and so you've got clean planting material that you take the plants out of tissue culture. You grow them in conditions where you know that are free of Fusarium, where you may then be establishing a nursery. But the important thing is to put them into clean soil, and that way you can control and stop the spread of Fusarium. So there are many potential sources of inoculum of spread of Fusarium. excuse my little diagrams, but pigs. And particularly that has been an issue for us we think it's been an issue for us in North Queensland, where we have had tropical race 4 come in. And there's lots of feral pigs going around in the national park areas. Some of them are adjacent to the banana properties, and they'll come in and they'll scuffle around in the banana plants and try and get hold of some fruit, and they'll just have all that soil on their hoofs, their feet anyway, and they can spread it around quite easily from one area to another. Gosh, what do you call them? Weevil beetles that can get in and out of corms. And I had a PhD student several years ago who did a study who could find, the Fusarium spores and part of the soil infested with the Fusarium on these beetles, and they're moving from plant to plant. So they're quite capable of spreading the Fusarium. Sorry about the bucket and spade, but that was to illustrate planting tools. Tools digging in, you're going into the roots, and you can get it on your tools, you can get it on your tractor tires, you can get it on your boots, etc., and you can spread from one location to the other. So it's so important that when you have got the potential of Fusarium being present, even if it hasn't been officially diagnosed, identified, is to maintain absolute hygiene going onto your banana properties. It's absolutely essential. And you will find in Australia now, on all the banana properties, have strict quarantine procedures and biosecurity procedures. So to get onto the property, you generally have to leave your own vehicle. You have to walk in. You have to walk through a footpath, and you might, in some cases, have to actually change your shoes, etc. If you want to visit your farming neighbour, you phone them and you meet them at the gate, and they'll maybe drive you onto the property using their own car. So it's very strict to try and minimise the spread of this fungus, because the issue is that once it gets onto the soil, it's there for good. Okay, so just please interrupt if you've got any questions. I don't always pick up on the chat, so I'm quite happy for you to call out. Oh, and cane knives to a certain extent, because a cane knife, the thing is that the infection has to get in via the roots. So cane knives you can cut the material. It could have spores on it, but it's unlikely to infect another banana by cutting with a cane knife. But the spores could drip to the soil and get in that way. We're not aware of infections happening via cuts, but that's not to say it could be possible. And then, of course, water. If it can get into the water course, and the spores will survive to a certain amount of time, not in flooding water, not in continuous water, but they will travel in water courses and down irrigation channels, etc. But the biggest spreader is us, human beings. They're very efficient at doing the wrong thing and of spreading it. So we have to always adhere to those biosecurity measures. So here in Australia, I talk about 3 races that matter. We talk about race 1, and race 1 is the strain of Fusarium Oxysporum that affected the Gros Michel cultivars. Cavendish is resistant to it. But we have other cultivars in Australia, which we call ladyfinger. I think they might be slightly different from ladyfingers considered elsewhere, but they're possibly a sweeter banana, not such a high yielding one, but they're susceptible to late race 1. We tend to grow them in the southern parts in southern Queensland and northern New South Wales. If you're familiar with the Australian geography, in New South Wales is where Sydney is, but it's only northern New South Wales that they tend to grow bananas at a commercial level, but not nearly as much as in North Queensland, where much of the production is. So we had subtropical race 4 come in. Now, subtropical race 4, not everyone's familiar with that. It was affecting Cavendish in suboptimal conditions. As I said, we grow banana here in southern Queensland, which is subtropical. It's not fully tropical, we're below the tropic, and we get, quite relatively chilly in the winter periods, and so tend to find that bananas are grown in these regions on slopes and to stop the buildup of any frost. And it's not nearly as high yielding as it is in North Queensland and the true tropics. But Cavendish sometimes succumbs to this race known as subtropical race 4. We don't know what it would do in the tropics because we've never introduced it, but these races that are form into subtropical race 4 are also known in South Africa as well. But it was tropical race 4 that was first identified initially it seems that it turned up first in Taiwan, but then in Indonesia and Malaysia, and it's been spreading since. We have it in Northern Territory in the 1990s, and just only 4 years ago it first turned up in North Queensland. It's now spreading worldwide. It's been in the Philippines for quite a while. It's spreading to Pakistan. It's in India now. It's in Mozambique in Africa. And just a couple of years ago, it turned up in Colombia in the main banana export production zones. And that's a serious concern to their industries there. So just to emphasize where bananas are produced in Australia and where we see it. So we've had race 1 here present in, there's a little bit of production in Western Australia, not very much, but we've had it present in Western Australia, North Queensland, way back since the start of the 20th century. It's been present there and in northern New South Wales. And now, as I mentioned, subtropical race 4, and more recently, we've got tropical race 4 in these regions too. Tropical race 4 is still under biosecurity. It's very much contained, not maybe a little bit less in Northern Territory, but definitely in Northern Queensland. I think it's only on 5 properties, and it's completely partitioned off in an attempt to stop its spread to other properties. At least give us time to work out ways to control it, whether through genetic resistance or through other means. So this is a distribution. I mean, this probably needs updating. This is from last year. It's probably spread since there. There's a rather bizarre indication that it's present in the UK. Apparently, it's in a glasshouse, a large glasshouse display at an exhibition. Somebody identified it there. How on earth it got there, I have no idea. So, right. So how to control this persistent soil-borne fungus? Yes, yes. Is there a question there? Yeah. No, I'll check in the chat. No, I can't. So anyway, maybe someone accidentally did that. Okay. So how to control this persistent soil-borne fungus? Hopefully you're all still seeing my presentation. In a monoculture of a single genotype, clonally propagated perennial crop, mostly grown in areas of high rainfall. So basically what I'm saying as a plant pathologist, you couldn't devise a crop that's more susceptible to a pathogen than banana. It's potentially a disaster waiting to happen, and it has happened. So if we're trying to control it, we can use quarantine, we can use biosecurity, as I mentioned before. We need to have effective diagnostics so that we can pick it up straight away and deal with it as quickly as possible. We can use cultural control to try and by knowing a little bit more how it's spread and how it exhibits its symptoms in the plant, we can maybe use some better control measures. And at the end of the day, I would always say resistance is the best policy. If we've got a resistant cultivar, all the best. So that's my little indications of how we're going through the talk. Now, I noticed in the little bio that Bintang provided you beforehand, there was a mention of VCGs. And I thought I might take a little minute to talk about VCGs. Now, please interrupt me if I'm going on too long. But I thought what I've often found when we've talked to not just growers, but to researchers and banana extension officers, and we talk about VCGs of Fusarium Oxysporum, they don't necessarily know what I'm talking about. So I'm just going to take a little bit of time to explain what a VCG is. Bintang, is that okay?
Okay, Prof. Liz.
Yeah, yes, you're okay with that. So, so if you want me to speed up a bit, or if you've already covered this in other lectures or talks, I'm happy to skip it. But basically, vegetative compatibility grouping is not just used with Fusarium oxysporum, it's used with a range of fungi, and it's to identify genetically related groupings. And it's a quirky thing that happens with fungi, and it's where the fungi will come together and fuse and actually can exchange nuclei. Now, they only will have this fusion only occurs between near related groupings. And it's thought that it doesn't occur on wider groupings between more distant individuals because it's some way of containing or preventing viruses spread through a fungal population. That's the theory behind it. But anyway, in practice, it's useful for identifying groupings of the fungus, and in those groupings, those that are related in their pathogenic forms. So, so what we do is that where they're incompatible, there's an inhibition of fusion between the mycelia of two different individuals, and you say these are incompatible isolates, whereas when the fusion occurs, they're compatible isolates. Now that's all very well. That's the theory behind it. How do you identify that? And you can identify it using a little bit of a trick in a cultural technique. And so we know that the VCGs of race 1, 2, subtropical race 4, and tropical race 4 differ. So they've all been given this international sort of numbering, and it relates to Fusarium oxysporum in general. Race 1 and 2 are sometimes interlinked, and so the distinction is less obvious. But subtropical race 4 have different VCG groupings from tropical race 4, and these are all very different from race 1. And in order to do a VCG test, it can take up to 6 weeks. And that's how it was done before the use of molecular diagnostics. So we're interested in tropical race 4, but particularly here in Australia, where subtropical race 4 is a little bit more prevalent, and you see a sick Cavendish plant, your assumption might be it's subtropical race 4, but you have to verify it's not tropical race 4, because tropical race 4 is so much more aggressive, and it's something that we really need to be able to stop the spread of. So hopefully my slides. How do we identify VCGs? So I'm just going to take a few minutes to go through this. Some of you might want to tune out if you're not that interested. But it's by complementation of mutants in the nitrate reductase pathway. So the nitrate reductase pathway has nothing to do with pathogenicity or what makes one race more pathogenic than another. We just utilize that pathway to identify vegetative compatibility groupings. And this was a technique developed by a chap called Pula, I think, way back in the 1980s or 70s, in fact. And if you just imagine the pathway in the fungus that leads from nitrate to ammonium. Now, Fusarium can utilize nitrate in media as a nutrient source by converting it to nitrite and then to ammonium. So if you've got nitrate in your medium, your Petri dish, the fungus will grow away, and it will grow. So the fungus to utilize it has to have effective steps in that pathway in order to do that. So if you then mutagenize the fungus so that it's no longer got effective steps in that pathway, it's not going to be able to get to the ammonium step and not going to be able to use nitrate in substrate. So what you can do is grow fungus on chlorate. Now, chlorate will convert using the same steps in that pathway. Sorry, this does get a little bit tricky, but go back over the slides afterwards if you're lost. Chlorate will go to chloride in the same way, using the same enzymes of the nitrate reductase pathways. But the thing is, chlorite is toxic. So if you grow the fungus on chlorate, it'll end up dying unless you've got mutants in the nitrate reductase pathway, and in which case they won't convert the chlorate to chloride and they'll still be able to exist on that medium. So that's what we do. We grow them on the chlorate, select mutants are able to exist on that chlorate medium, and we know that there'll be individuals that are lacking some of the steps in that pathway. And if we then, on minimal media with a sodium nitrate source, grow individuals where we know there are mutants in the steps in the pathway, and we grow them together, if the compatible groupings, if they're isolates from the same compatibility grouping, they will complement each other on that medium. And so it will be able to convert the nitrate to nitrite and then ammonium by flipping between the two different pathways to create it so that they can then grow. And so just as you saw this growth here, that's because the two isolates that were paired up together were able to exchange nuclei, and they were able to complete that biochemical pathway and make use of the nitrate in that media as a nutrient source by converting it to ammonium. So yes, it's a bit of a complicated process, and it can take up to 6 weeks. So if you've got a suspect isolate in a field that you think was tropical race 4, and you have to rely on this technology to determine if it is tropical race 4 as opposed to subtropical race 4 or race 1, or just even an endophyte, you have to go through these steps if you're relying on traditional techniques. And that can take time because you have to mutagenize the fungus and then et cetera, et cetera. So that's what we mean by VCGs. So what we now do is we run it DNA, and I'm sure most of you would be familiar with PCR now, particularly following COVID, where we had so many PCRs done on our samples. So you run a PCR polymerase chain reaction, so you're amplifying the DNA at a specific point, check it on a gel, or you might run it through a sequencer, prepare for sequencing and look at the sequencing. And you can do that. You can do that with where you actually sequence it, or you can look for a banded gel. You might use conserved genes for that. I will go through this quickly because this isn't a molecular biology lecture, but you can look at conserved genes, which allow you to look at the sequence, and you can make associations with those genes which you know to be associated with your particular fungus. And one of the ones that's used quite regularly with fungi as a barcode is the ribosomal genes, that we sequence the little in spaces between the protein the sections that lead to the proteins, and we can look for variations. So the more variation there is between any two individuals, the less likely that they're related. Okay, do we have a question in the chat? No. So just internal transcriber, these are used. So I'm just putting that up. We don't need to go through that today, but just so that you're aware of the background with a lot of these molecular diagnostics. And let's just, so we look at that. And you can take the information from this, because there are multiple copies of particularly the ribosomal genes. You can take the information from that and determine how related any two individuals are to each other. Sorry, I shouldn't have put in all these slides. We can look at them afterwards. Okay, and at the end of the day, this is what I was trying to get to. We can develop a tree of relatedness. So this has been done for many Fusarium Oxysporum species, isolates, and it's been done using several different techniques. And we tend to get a very similar sort of picture that Fusarium Oxysporum as a whole species can be divided into generally 2 or 3 very strong clades, main branches of that tree. And what we find is that race 1 and 2 tend to cluster reasonably close together, and tropical race 4 and subtropical 4 cluster fairly close together. And interspersed in that are other Fusarium oxysporums. You can get ones from strawberry, from tomato, and from melon, watermelon, sugarcane, or not sugarcane, what's that? I can't remember what that one is. Pea, ginger, etc., cotton. And they're all sort of mixed up. And then we've got the endophytes. Remember we mentioned about the endophytes, ones which will just live in the roots of plants without causing any damage. They can be interspersed in this tree. Now, this tree was just to focus on FOC, on the banana infecting ones, but we see them as quite different. Now, there's some recent proposals suggesting that there are actually Fusarium oxysporum should be divided up into several species. And I probably would agree with that. Certainly, 2 main species with the clade B and clade A, and this they're quite genetically distinct, but they look the same. The mycelia is the same. They sometimes the aromas they give off in media may differ. The spores are the same. The macrospores, the microspores, the chlamydospores, they're all asexual spores that they produce. And in the case of FOC, they all infect banana, but different races, different cultivars of banana. So there is an argument for, and there's also an argument against subdividing into different species. I just stick to Fusarium oxysporum because I'm used to it. So, but this does take me to another area of study where a lot of study of Fusarium oxysporum in general has been done on the tomato infecting strains known as Lycopersicum FOL. And this is work of a group led by Professor Rep at the University of Amsterdam, and where his group looked at the proteins that were produced in the xylem, in the water conducting vessels of the tomato plant when they were infected by the Fusarium oxysporum. And they've identified these little peptides, these small proteins, and knowing the protein sequence, they could then work back to find the DNA sequence that corresponded to that. And they unfortunately called these 6 genes as in secreted in xylem. But it turns out there's a whole range of 6 genes, and we now have 6-1 and to 6-14 types of 6 genes. So sorry, the nomenclature is confusing. So we wanted to look to see if these 6 genes were present in the banana infecting strains, FOC. And so initially we did southern analysis, and now then the technology moved on and we could analyze it by sequencing. And we found that 6-1, initially our initial studies showed that 6-1, 7, and 8 were in the banana infecting strains, and that there were different combinations depending on whether we were looking at subtropical race 4, tropical race 4, and race 1. So there was a means of distinguishing them. And so we published that work, oh gosh, it's 10 years ago now. And sorry, I should say, I'll go back. Should have said we can identify that basically by looking at bands on a gel. If we do PCR reaction, which is specific for each one of these 6 genes, we can look for their presence, absence on a agarose gel and compare them. So we could distinguish between the different races this way, which was a lot quicker than doing it by VCGs. But then we realized it was a little bit more complicated than that. So this is Liz Cieslowski, who also did a PhD in my lab, and she investigated the diversity of these 6 genes in the banana pathogens using lots and lots of different samples. She used a technique that was available at the time called MySeq. This sort of technology moves very quickly. But basically, what she's doing is looking at lots of little sequences that are generated from your sample and compare it against a known. And in this case, the known was from the tomato infecting strain, FOL, and looked for similarities and see if there was any variation in the sequence. And whenever there's a color there, it means there's a variation to the standard. So don't worry too much about the technology behind it. But basically, at the end of the day, she got a really nice pattern when we look at different VCG isolates and we group them into the different races that they're associated with. We could see a distinct presence and absence of whether it was tropical race 4 or subtropical race 4 or race 1. And the interesting thing is these 6 genes are producing proteins in the plant that are thought to be involved in pathogenicity. So this isn't just like a random association that we predict that they may be involved in how they attack the plant. It's certainly known in the tomato system that the 6 genes have been identified as the effector factors that interact with resistance genes in the tomato plant. So we see a nice pattern from this. And what Liz then did is she looked at it in a little bit more detail and actually looked at the sequences of these 6 genes. And we could see something quite interesting in that with the tropical race 4 ones, they seem to have 3 copies of what is known as 6-1, and 1 copy was completely unique, was not seen in any other FOC. So, so whether there's some feature of that, and a 6-1 is a peptide that seems to be associated with a lot of Fusarium oxysporums that are pathogenic. Many of them have that particular one, and it's thought to be a very important in pathogenicity. So maybe 6-1I is a variant that is something particularly unusual to tropical race 4, and maybe it's what makes it that little bit more pathogenic, a bit more aggressive. We don't know, we're still trying to work that out. But certainly other groups have worked on the 6 genes and identified various features about them too. So this was a way of distinguishing Fusarium Oxysporum f.sp Cubense tropical race 4 by identifying that particular sequence was another way. It wasn't just presence absence, we could get a little bit more specific. So these differences can be used as targets for molecular diagnostics. And as they have been done, there's a paper by Carvelis et al. In Frontiers in Plant Science, where they've utilized 6 genes and differences in it to try and use it as a mechanism of distinguishing the different Fusarium Oxysporum cubenses. I should add that this is an illustration that Liz did of the 6-1 variants, the sequence variations that you found in the different isolates. And as I said, tropical race 4 had a very distinct sequence compared with all the other FOCs. And that sequence seems more associated with strawberry infecting strains and pea and melon infecting strains. So there's indications, and other publications have indicated that too, that there's a certain low level of horizontal transfer gene transfer going on in Fusarium oxysporum populations. So whether the tropical race 4 has arisen because it's acquired a different pathogenicity factor from another Fusarium oxysporum, and that's what's made it more virulent. I don't know, that's just speculation at this stage. So so 6-1 is not found in any other banana infecting 6-1I, but similar sequences are found in strains infecting strawberry, pea, and watermelon. So that's quite intriguing. Okay. All right. So I've talked a little bit about the diversity of the pathogen, and now I'm just going to look at a little bit how are we doing for time by time. Do interrupt if you want me to stop, but are we going okay? No?
I think it's okay to continue it first.
Okay, all right. Okay, so how do we control Fusarium? To control it, we need to know where it is. And remember I mentioned how the most distinctive diagnostic is when you chop the plant open and see the vascular discoloration, because those other external symptoms can be associated with climatic conditions, with rain, or other fungi. So we need to have a little bit of an idea of where the fungus is and how to control it. So to what extent does the fungus colonize plants and where are the spores produced? Now, there's been several studies done looking at the actual infection process through the roots, but we were interested to see what happens once it gets into the plant. How does it spread and where does it go? So we know that the fungus itself can produce long-lasting chlamydia spores, or which are, for those that aren't familiar with fungal pathogens, what chlamydia spores are they, it's when the mycelia, when the fungus loses, when the nutrients are depleted and the fungus is basically its last gasp, it will cleave off the cells of the hyphae and form these resting spores, which are thick-walled. And so it's there for when a substrate, a medium comes along again, and it can germinate and grow and attack another plant. Or it can produce these macroconidia, these banana-shaped spores, which can spread from one plant to the other, or can be spread from one plant to the other. Or it can produce these little microconidia, and the microconidia, the smaller spores, are thought to move up through the xylem tissue. And there's some sort of classic studies were done back in the 50s and 60s, 1950s and 60s, looking at the spread of Fusarium Oxysporum, not just in banana, but in other things. But they didn't have the advantage then of having the material to work with, the instruments to work with that we now have. So we were interested to look at the spread of the fungus using a GFP transformed strain of FOC. Now, we had a subtropical race 4 strain of Fusarium Oxysporum Cubense that had been previously transformed by a PhD student of mine using the jellyfish protein, the green fluorescent protein, I should say, and from jellyfish. And so it has a constitutive promoter in front of it, so it means it expresses all the time. And then so we inoculated the plants in controlled conditions because it's a genetically transformed organism. We have to make sure it's contained. And then using confocal microscopy, we could then look at how it spread through the plant. And this is Nolene Warman, who conducted a lot of this work for us. And this is an image of, on the left, we've got a plant root that was inoculated. This is 20 days post-inoculation. It's the ladyfinger that has been inoculated with the fungus. And we see this mass of fungus network on the surface of the root, a bit like you'd see a microrhizae on the surface of the root. And so it's obviously it's gained entry from there. And then this one we see is 70 days post-inoculation. And this one really blew my mind away, that I had not appreciated how intensive the mycelial growth was in the xylem vessels. Now you can see it's the xylem because you see the little pits in the background. So you see that, it's the water conducting vessel. And you must remember that xylem itself is effectively dead matter. It's a lignified tissue. It's made up of lots of cells that come together but then died off. The living cells are all adjacent to it, but the xylem itself is like one big, huge straw that goes up through the plant with multiple. In the case of banana, you've got multiple xylem vessels all side by side. And so you can see that the fungus, the mycelia here, you can see a few spores, etc., growing up through the xylem. We look at this one. This is on the right. This is a Cavendish plant, 50 days post-inoculation. These are all glasshouse grown plants. They're not in the field. We can't take these genetically transformed fungi into the field. And the one on the right is a transverse section through a pseudostem, and you can see where the vascular bundles would be, where the xylem. And in most cases, they're actually clogged up with the fungus. And this, again, we see on the left-hand side. So this is subtropical race 4 on Cavendish. And so basically, this is what you're looking at. If you were looking at, and it's nicely marks where the xylem is in that section. And here's another one. Now, this is interesting because this is what we saw on decaying leaf sheaths. We think of this fungus as being soil-borne, but we actually saw aerial production of spores. Whether those spores would be not necessarily able to infect other aerial parts of the plant, but they would certainly fall down into the substrate and they could multiply there. And you see, so if we look at this image on the right, you can actually see the pincushion pustule, or whatever you want to call it. Forgotten the technical term, of the spores being produced at that point on the surface of the leaf. And here you can see all those macroconidia in that xylem vessel again. Interestingly enough, what we did see is that, maybe it's on the next slide, no, maybe not, that in leaves that were still quite green, that weren't showing any symptoms, the fungus had actually moved all its way up, and we could section the leaf and look at the xylem, and the xylem was full of spores. So even in a green leaf that was not yet showing symptoms of Fusarium, the fungus was present. So Fusarium Oxysporum is a hemibiotroph. So it can spread around in a biotrophic phase. I don't know what nutrients it's gleaning from the xylem, but it does do it before it actually spreads into the living tissue and sort of necrotizes it. So it's a very, very cunning fungus. And here again, we're seeing spores coming off on the leaf surface. So the kind of lesson from that is, quite often farmers or people who maintain a banana plantation will go in and de-leaf to get rid of other leaf pathogens of leaf spot, black sigatoka and things like that. But sometimes if you're de-leafing, you might actually have on those leaves that are falling to the ground masses of Fusarium spores, and then they break down in the vegetation below. So it's a way of the Fusarium still being maintained in the plantation, in amongst the bananas. And you can see here, we've seen chlamydospores, those resting spores, sometimes existing in the air chambers of the banana tissue. So the interesting thing is that Fusarium is confined to the xylem in healthy-looking leaves, and as the leaf senesces, the Fusarium then colonizes extensively as a saprophyte. So the implications are from that is that trash from de-leafing subsequently becomes a source of inoculum, including chlamydospores. And then we had Jay Anderson, who worked in the lab for a while, and she looked at what Fusarium did on non-hosts. And we could get a minimal level of infection in some of the weed species that we see, things that are known, sorry, I haven't got the Latin names, but things known as cobbler spelts and pegs, and some of the nightshade, which are in the solanaceous family, and Lucina, which is used as crop for cattle. We could see a limited amount of growth there, but not too much, not that it was going to lead to a buildup of inoculum necessarily. But when we sprayed with herbicide, and as is often will happen, and particularly happens in a plantation where you're trying to inhibit the spread of the Fusarium, you go in with a herbicide and kill the tissue. When you killed the tissue with the herbicide, the Fusarium took over and it went into its saprophytic phase and the inoculum built up. So it's a tricky thing to deal with. And even when we tried using in combination a fungicide and a herbicide, it didn't inhibit it. We still got a lot of buildup of fungus. So this is a fungicide drench trial on a banana infected plant. So we were trying, to try and eradicate it. And then when we sprayed it with the herbicide, you actually got an explosion of the fungus so that has implications for containment controls. And I know that Queensland Biosecurity and through working with the banana industry have strict protocols on how you attempt to eradicate it, and involves actually removing the plant tissue and treating it with ammonia and confining it in plastic bags and stuff, rather than just going willy-nilly and spraying with a herbicide, because that's not necessarily going to reduce the potential inoculum. Okay, so that's mostly what I want to talk about, morphology and diversity. I can talk a little bit about resistance if you would like, but we can stop there. Bintang, what would you prefer?
Okay, maybe if some of participants, if you have maybe questions,
Yeah.
you can directly or you can just put on the chat box. Do you have any
Right.
questions, guys?
Any questions? Because I'm going to sort of, if we talk about resistance, it's quite a different sort of area we're going to go into if that's what you want to hear. So
Yeah.
any questions? I should have inquired a little bit more about people's background, Bintang. So are we talking to students who in animal and plant science?
Okay, I think the most participant comes from another university. Also, some of them, I saw some plant pathologists too. And I
Right.
saw some of my colleagues from the plant quarantine.
Mm-hmm.
Another, yes, another researchers also here. So maybe if you have any questions, you can say it directly to Professor Elise. Okay, so this is Khairul Anwar.
How do we know?
Okay.
Right. Okay.
So we have
So
questions here. So can
right.
you,
So how do we know if our banana is attacked by FOC? So if you've got Fusarium wilt symptoms, which, and particularly that vascular discoloration, and you've not had Fusarium wilt before in your plantation, I would imagine that's the time when you would call in for expert opinions from your diagnostic people, from your extension workers who will work with this. The important thing is trying to minimize the spread. So if you think it's potentially Fusarium Oxysporum, and if it's tropical race 4 and you've not had that before and you're wanting to make sure you don't get it spreading, you really have to isolate that zone where it is. It's very difficult. There's plenty of stories around the world of when it's turned up in Mozambique, when it came to Colombia. It's that attempting, but initially give yourself time to try and reduce the consequences of spread. Certainly don't take suckers or anything from that material. If you want a proper diagnostic, I'm not the person to tell you what happens in Indonesia for that, but certainly I'm sure there are some labs that will be able to assist you, whether they're using the traditional BCG technology or whether you're using diagnostics based on molecular assays. They should be able to confirm with you whether it's tropical race 4, subtropical, or it's unlikely subtropical race 4. Or in some cases, you will isolate Fusarium oxysporum from a banana plant regardless, because often that's endophyte, but you have to determine if it's a pathogenic strain. And so I guess that's the best thing. But it's just, even if you don't have it in your plantation, I think it's good practice to have biosecurity measures. And I know my colleagues in the Department of Agriculture and Fisheries through ACR projects working in Indonesia have hoped to pass on their experience with dealing with tropical race 4 in Queensland to dealing with it in Indonesia. So by using quarantine practices where best you can, don't go into a clean plantation with boots that you used in a dirty plantation, for instance. Always just think of ways that the fungus can spread, and it can so easily spread. I know that one of the international researchers in demonstrating it, he was from the Netherlands, he went to the Philippines, and he got back to his hotel. He took some scrapings from his shoes and cultured it, and sure enough, he was able to get tropical race 4. So it's so easily spread, and just think of ways of minimizing that spread. Yeah. Anything else?
okay, to
Oh, you'll have to translate for me, please.
oh my God. So this is from Basuki. So is there any way from VOC to infect except from the roots?
Far as we know, it's interesting because you can isolate endophytic Fusarium oxysporums quite high up in a plant without any indication of how it's traveled there. I think all the evidence so far shows that they come in via the roots, by the root tips. course, obviously, if your rhizome is already infected and you've used that as a growing material for a new plantation, you're going to spread it that way. But it tends to, from all the evidence so far, it gets in via the roots. We tried doing a few experiments to see if we could do some stem inoculations to see if it would spread from that. And we did get localized growth in the stem, but it didn't seem to get into the vascular tissue. So I think we can assume it's mostly by the roots, but there's always exceptions. Can we use antifungals that we can find in the market? I know that there has been work in India using trichoderma species to try and control it, and they've got some reasonable results. And sometimes it's a combination of different things. With tropical race 4, it's so aggressive that if it's there in high inoculum levels, you won't control it. But you can use a combination of different things. We've tried using silica, applying calcium silicate to try and make the plants a little bit more upregulate the defense mechanisms. We've also looked at using non-pathogenic Fusarium Oxysporum strains, and there's certainly evidence of that being effective in tomato, where they use endophytic Fusarium Oxysporum. It seems to upregulate defense pathways and outcompetes the pathogenic strains. We've seen evidence in pot trials that it does have an effect, but again, in tropical race 4, if it's so, it can be so aggressive. You know, so there are various things that you can apply to the soil where it can be trying and outcompete the Fusarium, but it's maybe a combination of both of many things can curtail it, can slow it down. I know that Syngenta, I think it was, did think that they had a chemical product. I think it was something, it was I'm not quite sure what type of thing it was. It was maybe one of those chemicals that upregulates defense compounds, but it wasn't completely effective. That's as far as I know at the moment. Certainly there's a lot around the world, and particularly the big banana producers in Central America are very keen to find something. Okay.
Okay, so this is still from Basuki actually. So how long the incubation from the first infection until it shows the symptoms on the banana?
Okay. Well, it's very weather dependent. I mean, a lot of our experiments have been done in pot trials in glasshouses. So I wouldn't necessarily say that's the best indication of what happens in the field, but certainly in our pot trials, that when we inoculate the plants, and we inoculate plants that are 3 months post tissue culture, and 3 months later, you will see very strong symptoms externally. In a plant growing in a healthy plant growing in a plantation, it could be a similar period of time. It may take longer, it may take less. Certainly in the field trials that they've been doing in Northern Territory, where they've actually inoculated tropical race 4 into the ground there, they can see symptoms in some lines very early on after planting. In other lines, it may take over a year. So it varies. It varies, and it varies in the weather events too. Sometimes there's an argument that dry weather followed by wet weather will cause symptoms to, be more obvious. It varies. Yeah.
Okay, Prof. Liz, is there possible more than one race can attack at the same time at the one plant? Is that possible?
Yes, and that's interesting because there's evidence. I don't know if you may have heard of Ken Pegg. Ken was still is a wonderful plant pathologist. He's well into his 80s here in Queensland. He works for the Department of Agriculture, and he always said that if you had race 1 in a field, you were less likely to get subtropical race 4. So there was an indication that race 1 gives on Cavendish, which doesn't normally cause any disease in Cavendish, will actually induce a defense response and actually make the plant a little bit more resistant. And I think there was a paper that came out recently, the Dutch group, Gerd Kemmer's group, I think it was indications where they showed that. And again, we've seen that we can get upregulation of defense genes with race1, which may make the plant less susceptible to tropical race 4. We certainly, it would be interesting to have a little bit, we've never been in a position where we can compare subtropical race 4 and tropical race 4, because we're not allowed to work with tropical race 4, and we're not allowed to take subtropical race 4 up north. So we've never been able to do it in the same plant. But certainly, the other interesting thing is there is other Fusarium oxysporums in the plant. There are the endophytic ones. It depends. It doesn't matter if you look at banana, you look at ginger, we see the same thing. There are other Fusarium oxysporums in the plant. I was quite alarmed initially when I had an honours student doing a glasshouse trial, and she was isolating Fusarium oxysporum from all our controls. And I thought, oh, how have you contaminated your controls? But it turned out they were all endophytes. So they will be there quite often. They're there. But whether you get two pathogenic strains in the plant at the same time, I can't see why it wouldn't be possible if they were inoculated, and you could see that. But I suspect that tropical race 4 would outcompete subtropical race 4. Yeah.
Okay, so we have some questions here probably. So, Haram.
Right, there's one here. I'm reading it here. Appreciate for this. In regards to tropical race, is there any new information progressing while? Oh, we need to go down. While we are worry about it, do you have information on co-infection of some races? All right, so that was the question we just had. Can you please elaborate more on subtropical race? Do they actually race for? Does it need a predisposition? You know, they talk about the predisposition. Certainly, we do subtropical race 4 inoculations in pot plants in the glasshouse, and we see symptoms. I have a field trial in a little bit of land here in Brisbane near the bay, and somehow, I don't know if it was our dirty feet or whatever, we managed to get subtropical race 4 into that field, and the plants got infected. The susceptible plants got infected. So it's the idea that I think Randy Platts from the University of Florida has published a bit on this, and he talks about it's an issue. It's where the plant is predisposed because you're growing banana in suboptimal conditions, or not as vigorous as they would be in the tropics. But I've never done the side-by-side tests. I suspect I would be surprised if they didn't get infected in the tropics, but I don't think anyone's actually done that. But maybe the plants can outgrow the pathogen to a certain extent. And sometimes you see it where you may get the first pseudostem, when you first plant, you maybe get the first plantings affected, and then it will grow out of it. The subsequent pseudostems don't get it. So yeah. Mention about the 6-1. Do you think this is most specific to FLC diagnostics? I having said that, because I think traditionally, well, not tradition, but a lot of diagnostics are based on the ITS. I flipped through that. I didn't go through that much in my talk here, but the ITS is the region in the ribosomes which goes from a highly conserved region into a variable region, and it's used as a fungal barcode basically to distinguish different species and stuff. And it was initially used as a diagnostic in Fusarium and tropical race 4. Other groups have developed a diagnostic. I'm a little bit concerned because I have seen the variability of Fusarium oxysporum and the fact that endophytes are present in the plant. And some of the diagnostics are based on directly doing a PCR reaction from DNA extracted from plant material, not going through culture. And the problem with that is that you could be picking up endophytes, and you could be picking up a random endophyte that's a very similar sequence to the pathogen, because you're looking at a region in the fungus that has nothing to do with pathogenicity. So I have a little bit concerns that you focus on one diagnostic based on one region. I always feel it's a little bit safer to look at different aspects, to look at the 6 genes by all means. But even then, you can see that the 6 genes can be moved around by horizontal transfer, albeit it seems to be at very low levels. So I think it's always safer on a diagnostic to target 2 different regions. Maybe do an ITS plus a 6 gene or something else. Particularly if you're going to do your diagnostic directly from plant material and not grow through culture, so that you know you've got pure culture. Because if you're doing it from plant material, there could be multiple different fungal strains there, and you might get the ITS of one thing and then you get something else of another one combining in your PCR. So yeah, so I think the 6 genes are a good target. But I think it's always best to use, if it's so important that you're going to go into a plantation and destroy everyone's plants in order to try and stop the spread of the fungus, you need to be 100% sure that you've got the right thing. And it's easy enough to run 2 PCR reactions as opposed to just 1. But that's my personal opinion. Okay.
Okay, so maybe this is kind of technical questions. Dede is asking about what is your major challenge for your works, especially about the FOC.
FOC.
Yes.
My biggest challenge we have is because we can't work with tropical race 4. I would like to work with it, but I wouldn't dare have it here, because I wouldn't want the risk if it turned up in Brisbane, they'd say, oh, those people at the university brought it in. So I think it's a bit of an issue that we can't actually work. We have to send it. We've recently sent samples to South Africa, to the University of Stellenbosch, for our actual plants, for them to screen them with tropical race 4, because we can't do that here ourselves. There is a site in Northern Territory, but it's because the Northern Territory gets such variable weather conditions, goes from dry to wet to monsoon, it's difficult to get consistent results. So so that's our biggest problem, what we do and just a quick answer to the person about long-term storage. Fusarium, for those of you who are mycologists would always know that Fusarium tends to lose its pathogenicity when stored in culture. A lot of that was thought to be due to transposons, but maybe it's due to the presence and absence of 6 genes. Once you grow something in a saprophytic condition, growing it in media, you may lose its pathogenicity. So we tend to store our isolate. The best way we store it is either lyophilizing it and storing it in -80 freezer, or storing it, growing on minimal media and storing it underwater in a cold room. Okay.
Okay, so maybe this is the last question for this session first. Okay, so from Widaryati.
So, are there genetic derivatives can affect the inhibition growth of plant? Are there FOCs that have minimal inhabitation? Because as long as I know, FOC can inhibit. It can inhibit plant growth. Yeah, is that what you mean? So it can reduce the plant growth. Yes, I mean, if it doesn't kill the plant, it certainly it will slow it down if it's a pathogenic strain. I know that from our plot where we had subtropical race 4 present. Even though it might not have been killing some of the plants were very much reduced in growth, but maybe it was due to partly to the continuous cultivation that we were doing as well. Is that what you mean? Sorry, I'm not quite sure.
Okay, so maybe Widaryati, can you explain more about the questions?
No.
Maybe how to control the growth of the Fusarium itself.
Yeah.
In the plant, maybe.
Yeah. So the plant is trying to inhibit it by killing off xylem vessels. Just as in a hypersensitive response, when at a cell level, a plant tries to control a biotroph by killing a single cell, and stop that fungus from growing. It does the same in a xylem vessel. It will try and kill off that xylem vessel to inhibit growth. That's the theory behind it. How much in practice? I mean, that's why I said I was really surprised when we looked at our GFP studies and we saw how extensive the fungus was in the xylem at a time when the plant wasn't showing any external symptoms. So so it obviously wasn't inhibiting its growth in the xylem.
Okay, sorry.
Okay.
Okay, sorry. Okay. So maybe honestly, it's kind of complicated about Fusarium Oxysporum itself because it's one of the pathogen with high diversity from morphology, even from the genetics. So yeah, maybe like Prof. Ellis said before, we need to find maybe more diagnostic tools. So later we can find what is the best method, what is the best techniques to control this pathogen, especially in field. Okay, so maybe you can continue your presentation first.
Okay.
Then we'll have a discussion later.
Yeah. So what you're still seeing it on the screen? Yes.
Yes,
it's still there?
very well. Yeah.
Okay, so what I just wanted to talk about a little bit about the genetics of resistance. Now, I did mention before there are breeding programs. We don't have any breeding programs in Australia. Our industry is too small. It wouldn't be economically worth it, but there are breeding programs around the world. Traditionally, there was one in Honduras that's produced a few lines. Then IITA in Africa, I think it's Icaria in India, and in Brazil, and the French breeding programs too. So we can breed. So banana, we know banana's a bit of a freak. It's sterile, it's a polyploid, it's parthenocarpic, it produces fruit without seed, which is no benefit to the plant whatsoever. And as a consequence, we have to clonally propagate it, otherwise it wouldn't propagate itself. Not the cultivated ones would. And we know that there are certain hybrids between Musa acuminata and bulbiciana. And if you see fruit, and I know that in Indonesia, some of your local varieties will actually have some seed in them, but generally not to the extent as this one here on the right, where you've got quite extensive. And we've been working on a line malacensis, which again, I think it's more endemic to Malaysia, but maybe extends into Indonesia. These subspecies of Musa acuminata. And as a diploid, it's a very skinny plant. And it produces seed. Now, the nice thing about this particular line, it shows segregation for resistance to Fusarium. Okay, so resistance is present in some wild seeded diploid lines, but obviously we need to get that back into lines that are otherwise desirable, are parthenocarpic and sterile. So. And also bananas, a long regeneration time, large size of plants, interploidy crosses, which make the genetics a bit messy. So the Musa acuminata we worked with produces small seeded fruit, no commercial value, which showed resistance amongst some individuals to tropical race 4, subtropical race 4, and also race 1. So the fact that some is good because it means that we can generate populations and look for segregation of resistance and try and identify the resistance that way. Now, malacensis has been used in a lot of the breeding programs in the past, not necessarily this malacensis, but other ones, some known as Pahan, and introduced traits, not just they didn't know it was tropical race 4 at the time, but introduced other characteristics. So we did some self-pollinations. We do embryo culture, so we would take the seeds, but we actually manipulate the seed, pull out the embryo, take it into tissue culture, and that way we can multiply up clones from an individual line, so we can, without going out into the field and back in through tissue culture. So it allows multiplication through tissue culture. And so we self-pollinate. By self-pollinate, obviously we don't do it on the same flowers because of the way that the male and female flowers develop. So we have to have two pseudostems of the same clone, where we can take the pollen from one flower where the male flowers have been produced and take them back onto the female flower. And then we can get seed from there. And that can take, from pollination to actually obtaining seeds, ripe seeds that are ready to culture from, it can take 8 months in our conditions in Southeast Queensland. So from doing that, they would then generate populations of plants which we screen against Fusarium. Initially, we screen them against subtropical race 4, and we've gone to the Northern Territory in the past and screened them against tropical race 4. And we're not the only ones to be working with these populations of plants. The Department of Agriculture and Fisheries have previously put the parental plants that we have into the field, and they consistently show the same results. The resistant lines are very, very resistant. Susceptible lines are very, very susceptible. So it makes life a little bit easier in that respect. So we then take the DNA from bulks of resistant lines and bulks of susceptible lines and compare them. So that by doing that, by taking from many different plants, you're sort of trying to neutralize all the other features other than resistance, so that you know that your resistant lines, the bulk DNA from resistant lines, will potentially contain the resistance gene and the susceptibles won't. But they'll be variable for all other genes or anything else in the genotypes. So we inoculate in pots, and we get quite distinct phenotypes depending on resistance, susceptible. Because we inoculate at such a high level, the susceptible, sometimes you get a little bit discoloration in the base of the rhizome, but that's all. Whereas the susceptibles are just on brown black, basically. And from doing that, and by comparing using a protocol known as SNP, single nucleotide polymorphisms. So those of you who are more familiar with molecular biology may know what I'm talking about, but scanning the whole genome of these bulks and looking for polymorphisms, differences in just very subtle differences in sequence. It's not necessarily anything to do with genes, it's just that the sequence as you read through it. We were able to identify a region in chromosome 3, which was seemed to be carrying very strong for the resistance. The difference between the resistant bulk and the susceptible bulk at that point was very strong. We're able to do this, and we compare it because it's just wonderfully. It's wonderful to see how much international collaboration and input goes into a lot of these things. And we were already able to compare it with the sequence that had been published by the CIRAD group in France of what they call the double haploid Pahan, which is a Malacensis related line. So a lot of what we do has been made possible by what other people do, and making that information publicly available. And I think it's easy and nice to look at. We could see these differences. So if we go along and we look at lines that we predicted to be homozygous for resistant, they were all predicted on the basis of the target regions that we were looking at, molecular markers. We saw that they were all very much resistant when we challenged them in glasshouse experiments. Were the ones that were predicted to be homozygous susceptible based on the molecular markers we identified, they were all very variable. And then those that were heterozygous, again, they were all resistant. So we could see disease amongst that. And another thing when we do is when we're looking back at that chromosome 3, I think this really illustrates that this is a distribution of SNPs, of these single nucleotide polymorphisms, comparing the resistant bulk and the susceptible bulk. And we see this very intense peak, and this corresponds to chromosome 3. And so this is the region of diversity between the resistance and susceptible bulks. So we're fairly certain that that's where the resistance gene in these populations lie. But it's not that easy to get there. So we're still trying to get there. But because banana is very much a poor crop in regards to crop plant science, if you like to call it, very, you know, and when we look at it, because there's a lot of introgression crossing over, it's difficult to have actual pure, you know, lines which are homozygous. And so we see a lot of diversity, and it's quite tricky. So the region we're looking at, to cut to the point, is very tricky. There's a lot of repeat sequences and stuff, but we're quite convinced the resistance is embedded there. So we're looking at resistant Malacensis. When we sent our DNA to a group in the Czech Republic who are very good at doing looking at banana genomes, but also looking at genomes in general. They look at chromosome counting and stuff like that. They could see that our Malacensis resistance was slightly different from the susceptible. So the segregation going on, that the diversity of our lines is not a single genotype by any means. And that Pahan is the double haploid that the French group will use. So it's still quite a little bit distal from that. So it means that we're not necessarily always going to see the same thing as what's been published in the sequence database. But we have developed, and I say I don't do very much. Most of this work is by Andy Chen and my lad, who's been here with us for at least 6 years, I think now, through funding that we've got from both the Australian banana industry and also through the Gates Foundation, through IITA. He's developed molecular markers that are associated, and all the time he's continually improving those markers as we get closer into the gene. But that he's initially by doing a PCR followed by restriction enzyme digest, he can see bands that are consistently associated with resistance. So if you look at this particular marker that he developed here, the top band is you see with the homozygous resistant line. So the line 12 would have been homozygous, carrying homozygous for the resistance gene. Line 11 would be homozygous susceptible. It's got the lower band, and where you've got both bands, it's predicted to be heterozygous. And the benefit of molecular markers is that you can go into a breeding program and use these without having to screen the plants with the pathogen. So if you're trying to develop ahead of time of whether to get plants that are resistant to tropical race 4, you don't necessarily want to introduce tropical race 4 into your location to do the screening. So you can use a molecular marker to predict the lines that are likely to carry that disease resistance gene and use that. And you're not just breeding for resistance, you're breeding for other characteristics as well. There's no point having a banana plant that's resistant but has no decent fruit. So, but you can skip, you don't have to go through that period where you have to inoculate plants and look for symptoms. You can just select based on the marker. So marker has been used in international breeding programs to select genotypes, of course, misspelled, carrying resistance in the absence of pathogen screening. And our colleagues in France, well, not so much in France, but but from the French group CIRAD, have used it, also been used at IITA and in Embrapa in Brazil. But it doesn't, the thing is, I should emphasize, it doesn't identify all resistance, only lines with a Malacensis background. And that's, always, you might think, oh, that's pity, it's not picking them all up. But on the other hand, that's good, because that is potentially indicates that there's more than one type of resistance to Fusarium wilt available, which is good, because at the end of the day, those of you who are a little bit more familiar with breeding techniques, you can pyramid, you can add the resistance genes from different sources into a banana plant all at the same time. That way, it's going to be a much more stable resistance. It's going to last through generations of the fungus. So, having said that, Malacensis, if you look at this wheel of diversity of banana lines, which includes both cultivars and wild diploids, we can see Malacensis sits here. Cavendish is way over there, so it's quite distant to a certain extent, although there's characteristics in Cavendish that are thought to have a Malacensis background. Calcutta 4, which is a known resistant plant, it's heavily seeded. produces lots of seeds. It's resistant to Fusarium. I think it's also resistant to leaf spot, but it's, agronomically, it's a useless plant. You could never grow it for fruit. And Pisanjaraboya is another one that has good resistance to Fusarium wilt. It's quite distally related to Malacensis. So potentially we could have at least 3 different sources of resistance. So that at the moment, what we're doing is we're doing further crosses using Malacensis and using a source of Pisanjaraboya to see if we can get segregating populations and then determine where the resistance is in the chromosomes. If it's not on chromosome 3, there's a good chance it's a different type of resistance. And even if it is on chromosome 3, it might still be a different resistance. So that's where we are at this stage. Gosh, hopefully I haven't bamboozled you with all too much detail. Well, there's something in the chat, is there? I'm happy to field any more questions, and I might just finish saying that I've been working on this for many years now, too many years possibly. And through it, we've had some wonderful collaborations and still have some wonderful collaborations with people across the world. And this doesn't include everyone, but from different groups, and that's what makes it very, very good. I do note we don't have any collaborations. My group doesn't have collaborations in Indonesia, but I know my colleagues at the Department of Agriculture and Fisheries do have, and we're certainly very happy to interact with people in Indonesia and particularly students who are keen to learn more. Yep. Thank you very much.
Okay, that was really interesting actually. Okay, maybe if you have any questions, you can put on the chat box, or maybe you just can simply raise your hand. Okay, Prof, please, you explain about the plant resistance. So maybe the resistance of plant is the best method or the best solution to control the disease for some area maybe. How about the use of chemical substance such as fungicide or herbicide or maybe fertilizer to control the Fusarium itself? What do you think about that?
I think if you can get a chemical that can do it, that would be good. But there's not much history of effective chemicals against Fusarium Oxysporum in any crop. We see we have Fusarium wilt in cotton, and it's not controlled by chemicals. It's being controlled by breeding, by having plants that aren't completely resistant, but are tolerant to the Fusarium. We see it in strawberry, again, it's genetic resistance that's been most effective. Tomato, again, it's genetic resistance they use to it. I know that some of the companies, I think Syngenta did have a potential chemical. I think maybe more something that upregulates defense is more likely to be effective. But because the Fusarium is in the soil, you would have to try and control it in the soil. You would have to use it as a fumigant, and soil fumigants are horrendous things because they kill the whole microflora. So, and then, the Fusarium would have an advantage in that there's nothing else controlling it. I think if it was a chemical, maybe a systemic chemical, something that's in the plant might be effective. I know my colleagues are looking at things like RNAi, sort of trying to target through topical RNAi, not necessarily transforming the plant, but topical RNAi, where it is applied in a way that it can target certain features of the fungus when it tries to replicate or tries to grow. Those are early stages. It might be effective, but again, it's getting it into where the fungus is. If it's getting it into the xylem, you would have to target, if for instance, it's the same way with the chemical. If you've got a systemic chemical in a plant that is infected by the fungus and the fungus is colonizing the xylem, there's no transpiration going on. So how is the chemical going to actually move up through the plant? So it's certainly very tricky. I think it comes down to, maybe I'm beating my own drum, but I think genetic resistance is the ultimate, whether that's through conventional breeding or through transgenics. I can't see, personally, I don't see a problem with transgenics in banana. It's a sterile crop. You introduce a gene into it, particularly if you're introducing a gene from another banana line, you're just circumventing breeding procedures. That I can't see an issue with it, but we have to make sure that the community is aware of what we're doing and that's accepting of it. And so that's probably the biggest stumbling block at the moment. With genetic, and you don't necessarily need to take a resistance gene. You can manipulate defense genes as we don't do that, but I know that some other groups are doing some things like that. And I know here in Brisbane at the University, Queensland University of Technology, they do a lot of work in that area. So with transgenics, so there are other potential ways. Oh,
Okay,
gosh,
so we have more questions here. So this is from Diva. Is that possible for FOC associated with other plants around banana plantations with Fusarium wilt disease? Because yeah, it's soil-borne pathogen. So is that possible?
Is it possible? Right. So I noticed you're not using odor acid. So yeah, and I accept that that's tropical race 4 probably is another species, but the taxonomists are still fighting over that at the moment, and I'm keeping out of their argument. I know there's certain groups of taxonomists who want to revise the whole Fusarium Oxysporum, not just on banana. So they're waiting to do that. So that's why I'm still calling it Oxysporum. But how about instead of Oxysporum tropical as a distinct species? I think I think at least that clade is a distinct species where you've got tropical and subtropical. I think it is different from the race 1 and race 2s. Where you put the line as to where the species is, I'll leave it for others to argue that one. It's difficult how to define a species when you're dealing with an asexual fungus, because in the old-fashioned terms of the species is something that will integrate with another individual. They all belong to the same species. But if you're dealing with an asexual fungus that doesn't go through a sexual stage, it's where do you define where species is? Anyway, I'll accept what the taxonomists come up with, so I'll leave them to that. We've got a very eminent Fusarium Oxysporum scientist here in Australia, Professor Brett Summerall, who's the curator of the Royal Botanic Garden in Sydney. And I'm waiting to see what his opinion is, and I'll go with that. So, but going back to the fact that you can get it in, it's host specific in that it causes disease on banana, but you will get FOC, tropical race 4, or what odoratism, if you want to call it that. It will go into the roots of other plants, but not cause disease. Was that the main question? Sorry. Yeah.
So this is
all right. What was the change premature? I don't think I can see the reasoning behind that publication, but my concern was that they just focused on the Cubense, on the banana infecting strains. I think the whole Fusarium Oxysporum complex, if you want to call it, needs to be revised, and I think we need to see what's going on. It's very confusing. I have a student working on ginger, and she looks at Fusarium Oxysporum, and it's called Fusarium Oxysporum f.sp Cubense zingiberri. And when we look at the 6-gene profile in that, it's very similar to other Fusarium Oxysporums that affect date palm and that affect alliums. And genetically, I don't know how different they are, but they certainly won't. You can't get a date palm 1 to infect ginger, etc. Although there's lots of similarities. So it's very complicated. But yeah, I'll leave other people to argue that one.
Okay, I also agree with that, Prof. Liz. It's kind of complicated to determine for the asexual fungus. It's also the same with the Coletotrichum gliosporides complex.
Yes,
it's also complicated, right? It's
yes,
also
exactly.
the same with Rhizoctonia solani.
Yeah,
so
yeah.
we need to have like an anastomosis groups. Yeah,
Yes,
so
yes.
that's the way complicated. So that's why your work is still ongoing.
Yeah.
and I think it's kind of interesting if we can
Definitely.
discuss about that. Okay, so this is another questions from Mr. Basuki. How about the plant rotation to control the disease, especially
I
about
think,
the Fusarium
yeah,
wilt?
I think there's potential with that. Certainly not to, it will never get rid of it. But to reduce the inoculum. And I think there's certain crops that certain people are looking in that may have a better effect at reducing inoculum than others. I think you have to make sure it's not something that can live in the roots, that because it's when it's there and when that plant dies, and the fungus can turn into a saprophytic stage, and the inoculum will increase again. So if it's something that inhibits it in the roots in the first place, that would be a good idea. And there are a lot of things about green mulching. I think onions or leeks or something have been suggested, and maybe even mulching it in. I know that in certain conditions, it was interesting that they have this site in Northern Territory. Now, the Northern Territory climatically is very different from North Queensland, where most of our bananas grow. North Queensland is continual wet tropics. It's whereas in the Northern Territory, it's more monsoonal. So they go have a really dry period and then a monsoonal period. And they have this, what they call the death pit, where they actually inoculated Fusarium into the fields, and so that they could screen various germplasms there. And then they didn't do anything for a couple of years. I think the grant finished and they were doing something else. And they went back into that area, which had been more less left fallow for that period. And there was very little Fusarium present. And I suspect that in the dry time in the Northern Territory, it's very hostile to anything surviving in the soil. And so maybe that's when it reduced. Whereas I have a colleague here in southern Queensland who bought a property that had previously been, 50 or 60 years ago, a banana plantation that had been wiped out by race 1. And so the person had stopped growing bananas. There was no bananas on the land for all that period of time. But it's in the nice subtropical environment of southeast Queensland, where we get more or less, we don't get drought periods. And he put in bananas, he put in ladyfinger banana, and they all came down with Fusarium after 60 years of it not having any bananas in that field. So it can be incredibly persistent. Yeah.
Okay. Okay, so if you have any questions, okay, somebody want to ask questions, raise your hand. Mostly we answer the question by chat, or maybe they are too shy. Okay, so still any questions, maybe?
Anyway, you have my email, and if anyone wants to, and I mean, I've enjoyed this. I hope you have, and I'm only talking to you, Bintang, but I through the text I've met others, and certainly it's very interesting to know. And you should tell me what you people are doing in this area.
So I saw some of my colleagues here, as maybe some from another national university in Indonesia, and some also from the plant quarantine. Also some researchers here, and I saw some plant pathologists also here. So if you guys have any questions, so I think this is really interesting topic, because actually it's kind of complicated, but it's fun to discuss, especially about the morphology, genetic diversity, about the fungal disease, because there are a lot of diagnostic method developed actually from conventional or molecular method, but yeah, it's still really complicated to differentiate, especially about the pathogenicity from the pathogen, since like you explained before, VCG does not tell anything about the pathogenicity. Yeah, so is there any questions? maybe some of my students, you have questions or? Okay. Okay, I think all the questions already answered
Right,
from the
okay.
chat. Yes, even though just from chat. So, okay.
And I'm sure it's lunchtime with you now, is it?
Yeah, it's the lunch time. So, I think maybe we can continue the discussion not only by this virtual meeting, but later maybe on mail or another
Yes.
occasions. And as moderator, I would like to say thank you very much, Prof. Liz, for your kindness, for your consideration to become one of our speaker on visiting professor program at Faculty of Animal and Agricultural Sciences, Universitas Diponegoro. Thank you so much. We get more knowledge and insight, especially about the AVOC. And we really hope that we can continue the collaboration in the future. We can meet directly, not only by online meeting,
Yes.
but
No, that
maybe you
would be
can
good.
come to Indonesia to find the bananas here.
Yes.
Because we have a lot of bananas here, yeah, with a high diversity of bananas. Maybe some day you can visit Indonesia to do your work.
That would be lovely.
Yeah,
Yes.
thank you
Okay.
very much for your lecture and hope to see you soon. And I'll give it back the agenda to the MC. Thank you very much.
Okay, thank you very much. And thank you very much for asking me. I was very honored to present today. Thank you.
Thank you.
Thank you, Professor Elizabeth Aitken for the lecture and Aisyah Surya Bintang, SPMHC as moderator. We got more knowledge and insight. Thank you, and we are so grateful to invite you here. Now, we will close this visiting professor program. We really hope that it becomes useful for us, gaining more knowledge and benefits in future. Thank you very much for your consideration joining us today. See you again in another occasion. May all of you stay safe and healthy. Wassalamualaikum warahmatullahi wabarakatuh, and good afternoon.
Thank you very much.
Thank you very
Thank
much.
you.
Thank you very much.
Goodbye.
Okay, bye.
Thank you very much, Prof. Liz.
Thank you.
Thank you.